In the last video, we explored how we can engineer our immune cells into CAR T cells to fight cancer. But what if, instead of engineering cells, we used antibodies as bridges to connect immune cells to their targeted antigens? That's what we'll explain in this video: bispecific and trispecific antibodies. Before explaining the complex structures and unique mechanisms of bispecific and trispecific antibodies, it's essential to first understand their precursor, the monoclonal antibody, its structure and mechanism, then build from there to the advancing antibodies. Monoclonal antibody is a Y-shaped glycoprotein composed of 4 polypeptide chains, 2 heavy chains and 2 light chains held together by disulfide bonds. The key regions on antibody structure are 1. The Fab fragment antigen binding, which represent the antibody binding site. 2. Fc fragment crystallizable, interacts with immune cells to trigger an immune response. The structure of monoclonal antibodies got improved into containing 2 different antigen binding sites, each capable of recognizing and binding to an antigen, and that is bispecific antibody. So there will be 2 different Fab regions. To further push the limit of antibodies, trispecific antibodies contain 3 distinct antigen binding sites. Here the engineering is more complex. Generally, there are 2 ways of accomplishing this. Tri-Fab structure, meaning 3 Fab arms extend from central scaffold, or Fc fusion-based formats, which integrate additional binding domain into the Fc region. The therapeutic effectiveness of these antibodies is significant because they can target 2 or 3 antigens at once, and their high selectivity can help to minimize off-target effects. Let's dive deep into their potential mechanism and approved agents. Bispecific antibodies can be used as a bridge between targeted antigen and an immune cells like T cells or NK cells. An example of that is blinatumomab, which targets CD19 on tumor cells and CD3 on T cells. For autoimmune diseases, bispecific antibodies can be used to block 2 inflammatory pathways simultaneously. There are currently 12 bispecific antibodies that has FDA approval as of 2024, which included treatment for different cancer types. Trispecific antibodies can be used to recruit multiple immune cells to fight a targeted antigen to enhance tumor killing. For example, targeting a tumor antigen and 2 types of immune cells like T cell and natural killer. Also, they can be used in targeting 3 independent antigen for multiple immune drug-resistant cancers. The triple targeting or multi-pathway inhibition feature of trispecific antibodies will enhance therapeutic precision. While trispecific antibodies are a relatively new and evolving area of biotechnology, several promising agents are currently in clinical trials or preclinical stages. Now, as promising this might sound, the engineering and production process can be complex. Let's start from their base, monoclonal antibodies, and explain how bispecific and trispecific antibodies can be engineered. Antibodies are traditionally produced through hybridoma technology, which involve immunizing an animal, commonly mice, with the targeted antigen to stimulate antibodies. After that, B cells will be harvested and fused with myeloma cells, creating hybridomas. Selected hybridoma will be expanded, and antibodies will be purified and humanized. Later, recombinant monoclonal antibody technology came through, which involve engineering and expressing antibody genes in host cells using recombinant DNA. The process starts by targeting gene sequence that encode heavy and light chains of the antibody. Then these sequences will be cloned into expression vectors. Finally, these vectors will be transferred into host cells. The process ends here if we are designing monoclonal antibody. But for bispecific antibody, we need to co-express 2 distinct sets of heavy and light chain into same host cell. And for trispecific antibody, we will either incorporate additional antibody binding fragments or co-express 3 genes encoding heavy and light chains and additional binding fragments. That's all for bispecific and trispecific antibodies, but the journey doesn't end here. Up next, affimers, aptamers, and nanobodies, innovations inspired by the power of antibodies.